When an infection starts, the first white blood cell to respond is one called a macrophage, and this identifies the invading bacteria or fungus, eats it, destroys it, and then alerts the rest of the immune system. However, in the case of some diseases, like cryptococcosis, which is the one that we work on, the invading organism, which in this case is a fungus, is able to survive inside that white blood cell and kind of hijack your own white blood cells in order to spread through the body. We know that many white blood cells have a system to overcome this by throwing those hijackers back out, and that system is called vomocytosis, but we don't know how it's controlled. So really this research was all about trying to identify the mechanism that allows white blood cells to recognise and expel these hijackers. There are many diseases, not only cryptococcosis, in which the pathogen survives and causes infections by deliberately hijacking the immune system in this way. So if we can develop ways to manipulate this and encourage the white blood cell to recognise and expel organisms like this, we might be able to limit the spread of infection, not only for cryptococcosis, but also for other invasive pathogens that are a significant threat to human health worldwide. So the way we tackled this problem was to identify signals that white blood cells use to control their behaviour, and then one by one to disable those signals. And when we did that, we discovered one particular molecule called ERK5, which seems to regulate this process of expulsion. And we were able to demonstrate afterwards that you can manipulate this particular protein in different ways in order to encourage those white blood cells either to throw out pathogens better or to keep them inside and try and kill them for longer. In the short term, probably the biggest consequence is that we have understood a lot more about a really subtle and new aspect of the human immune system. But longer term, obviously, our hope is that we'll be able to develop therapies that target this process. For example, you could imagine developing drugs that might be able to limit an infection so that it doesn't stop the infection altogether, but it prevents it spreading from your initial site of attack. Firstly, we'd like to broaden this out, not just from the one disease we've been looking at, but to see how much this process also plays a significant role in other major human diseases. And secondly, we're very intrigued about developing new ways to target that particular pathway in white blood cells in order to ultimately lead, we hope, into a better therapy.